Let me first thank Hartmut Neven for the invitation and Google. This is, the whole field is very exciting. I think the way that Hartmut presented it was spot on. I would like to make two comments on his intro. One is, this is perhaps half frivolous, but there is no absolute proof that the human brain does not factor large numbers, okay? And in fact, people like Ramanujan would be considered evidence to the contrary. So I guess what I'm saying is that we know so little about how the brain works that we can't rule out that it actually does things by brute force, okay? And the second thing is that I believe, and this addresses Hartmut's comment on Stuart Hameroff, I think that we're in the age of guesses here. We're guessing. People are putting their money on certain hypotheses. I personally think the brain is a quantum computer. If it can be done at all, it will have been done by the 4 billion year R&D program. And I think Stuart was the first to basically state that guess, which is still a guess, but I think it's correct. And we're working on hunches here. But every scientist worth a damn has to have a hunch. And I think that hunch, which was Stuart's contribution, will turn out to be correct. Okay? So let me see if the events prove me wrong.
Actually,
now,
it was Penrose's idea about the quantum, and I added the microtubules, but thank you.
okay, Penrose Hameroff, sorry. Now, going back to Mohan Sarovar's categories, he had level 1, trivial, level 2, semi-interesting, and level 3, interesting. My talk will be firmly stuck at 1.2, okay? Just barely rising above the trivial. The thing is, not being a quantum mechanics specialist, I find even the trivial really interesting. You guys have been living such a high life with entanglement that you've become completely blasé about the ordinary. But let me assure you, there's plenty of ordinary quantum mechanics and biology that deserves worrying about. This is the first time I give this talk, so bear with me if there are any sort of rough edges. But the work I'm going to talk about was done by myself in collaboration with two very good condensed matter physicists, Marshall Stoneham and Andrew Horsfield. And I'm a biophysicist, Marshall and Andrew are physicists, and we were dealing with a lot of chemistry. So when we ran out of chemistry, which was several times, we asked Roald Hoffman at Cornell, who very patiently answered some seriously stupid questions, and Abdel Rashday, who did some computations with Roald Hoffman. He was a postdoc there. So benzodiazepines, and sorry, I should mention that all the calculations I've done were done on SCM's wonderful software suite, ADF, which is a density functional theory software which is suited to idiots like myself, and the people at SCM are incredibly helpful. And of course, DARPA provides the energy for all of this. That's my email in case anybody wants to get in touch. So benzodiazepines. This is a picture of Leo Sternbach, who died 2 years ago, who discovered benzodiazepines accidentally in the late 50s. And he's holding in his hand, I haven't quite been able to figure out which one it is, because I'm not sure what that green atom is. Is it a bromine or a chlorine? But I suspect it's chlorodiazepoxide, which was the very first one, called Librium. And he was a wonderfully candid pharmaceutical chemist. It's very rare that you see chemists admitting to the fact that they don't have a clue and they just made it up. They basically wanted to have some fun with chemistry, and some of the products turned out to work. So this statement of his, our knowledge of the processes occurring in the brain was rather limited, I think is the understatement of the century. Their knowledge may have been limited, but they got extremely lucky. And in the early 60s, they discovered a string of molecules, including, for example, lorazepam. Now, I assume everybody in this room has taken benzodiazepines at least once. How many people here have never taken benzodiazepines? I don't believe that for a second. I don't believe that for a second. You probably have, and you don't even know it.
Generic terms, please.
Librium, Valium, Rohypnol, if you're lucky. Temesta.
Ask for another vote. Ambient.
Ambien. No, Ambien's different. Ambien is a non-benzodiazepine. Okay, let's have a vote again. I still don't believe it. Okay. You guys have far too clean a life. So benzodiazepines are extremely potent, remarkably potent molecules. They work at milligrams levels in the human body of 70, 80 kilos. They're sedatives, they're hypnotics, they're anxiolytics. The really powerful ones like Rohypnol, for example, which we'll come back to in a second, are a mixture of all these things. They can send you to sleep. They can make you completely relaxed. They're used as pre-op medication for most even minor operations. Some of them cause amnesia, which is midazolam, for example, makes you forget what happened in the previous half hour, which is, if there ever was a philosophical drug, that has to be it. You feel the pain when they've set your shoulder, and then 10 minutes later you've forgotten that it happened. Did it happen? Nobody knows. So the terminology is that's a nitrobenzodiazepine. You add you add a nice ring with an electron withdrawing group on the side. You add another methyl. And you end up with Flunitrazepam, which is Rohypnol, also known as roofies. What's interesting about the Rohypnol molecule, and this is the first thing that Roald Hoffman pointed out to me, was that there's a big conjugated system of electron bonds here that goes all the way up to the amide. Amide is not a double bond thing, but it's sufficiently conjugated. It behaves like a double bond in this kind of context. And so here you have a molecule which has the ability to carry electrons from one end to the other quite easily. And you'll see that this is probably important. So where do benzodiazepines act? Well, if you Google it, since this is the right place to mention Google, if you Google it, you will find these lovely drawings done by different artists. The benzodiazepines act on the GABA receptor. So let me just single out one of these fellows. And this is an artist's rendering of the GABA receptor in what I call squash notation, okay? In other words, all the subunits of the proteins are depicted as colorful volumes with some shading that look like the squashes you'd buy in supermarkets in this season. And squash notation in biology means you don't know how it works. Anytime you see that in a biology article, remember, that's an admission of ignorance, okay? And what's really interesting about benzodiazepines is that they work on the GABA receptor together with a host of other things. In this particular instance, you have on top left, you have midazolam, which is benzodiazepine, propofol, which I'm sure Stuart is very familiar with, which is a general anesthetic, pregnanolone, which is another general anesthetic, but a steroid general anesthetic. Ethanol, you're familiar with that. How many people have never? Okay. Isoflurane, and of course GABA. Isoflurane is one of the main inhalational anesthetics. And so these are a remarkable class of compounds. The one I've highlighted on the right, britazanil, is probably not very well known to any of you. It's a molecule that's been proposed as an ethanol substitute by enterprising British pharmacologists who believe that we should stop drinking booze because it doesn't do the job very well, and that we should actually actively develop an alternative to alcohol. This is a deeply unpopular notion, but I think it has a great future. And I just wanted to mention this wonderful molecule, which is a promising candidate to replace. I haven't tried it. And of course, all this stuff does have its usefulness. Now, how do we know these things work on the GABA receptor? People say, these drugs act on the GABA receptor. How do you find out that they do? Well, what you do is depicted here. You inject the RNA for the different bits of the GABA receptor into a frog egg, an unfertilized frog egg. You wait for a couple hours until it obligingly makes the receptors in its membrane. You put 2 electrodes, 2 micropipettes in the frog egg using one of these machines on the left. And you measure the current that goes through the membrane when you put on GABA, which is what the GABA receptor is sensitive to, and you get a trace. So the smaller of the two electrical traces is the current in response to GABA. Then you put on some benzodiazepines, and lo and behold, the response is increased. The receptor is responding to GABA and is responding to GABA more strongly when there's benzodiazepines around. And this is the fundamental finding, and this is what I'm interested in. I'm trying to figure out why the receptor cares about whether there are benzodiazepines around or not. So the GABA receptor is a membrane protein made of several subunits, and it sits with an exposed part at the top, which is exposed to the extracellular medium, and much of it is buried in the membrane. And it's made of 5 subunits, the alpha, then beta, alpha, beta, that makes 4, and then a gamma subunit. These are different proteins, and each of them comes in different flavors. You can have alpha 1, alpha 2, alpha 3, so you can, sorting out the GABA receptor has been the job of molecular biology and pharmacology for the last 20 years. It's a very complex receptor system. And the benzodiazepine binding site is between the alpha and the gamma. If there's no gamma, it doesn't respond. If there's the wrong kind of alpha, and we'll come back to that later, it doesn't respond. And that's a benzodiazepine on the same scale without the hydrogens being shown. So how do these things work? Well, the standard issue explanation that comes from pharmacology in general is that drugs work by a lock and key mechanism. So but what is meant by that is actually non-trivial. What happens when you put a drug onto a receptor is that it binds to it. And what you see here is a molecule coming in and the receptor binding to the molecule by 2 different same interactions. At the top, you might have a hydrogen bond, and at the bottom, van der Waals interactions. And the theory of lock and key interactions is that the energy of binding is used to turn the receptor on. Okay? That's where the energy is coming from. Okay? Something has to turn the key, and the energy for turning the key is the binding energy. So one of the things about that's important about lock and key ideas is that, of course, the potency of a molecule should somehow have something to do with its shape. If it's a key, you got to turn it, and, it has to have the right shape. Unfortunately for us, or perhaps fortunately, the way that Leo Sternbach engineered, so to speak, benzodiazepines was to have fun as a chemist. And therefore, he put in as many modifiable positions on the benzodiazepine, because what he wanted to do was synthesize thousands of compounds. That's what chemists enjoy doing. And so you have all these positions on the benzodiazepine ring that have been explored. And what is remarkable is that not much sense, really, has come out of this vast pharmacology as to why one particular one works and another one doesn't. So let me just go through that. First of all, in order to illustrate structure-activity relations, I've used a thread on a Russian do-it-yourself drug chemistry site called Blue Light. Those guys are clearly really good, okay? Because this person says, anybody know how to make a better benzodiazepine, basically, as if you're going to do that in your kitchen, and he gets the following answer. All right, and this is as good a summary. This is on a thread. This is as good a summary of structure-activity relations of benzodiazepines as you will find anywhere, and better than most, okay? So clearly these people, somewhere in the middle of Russia, are worrying about making a better Rohypnol at home, okay? I find that the web has revealed all sorts of things. So you can do all manner of stuff to the basic benzodiazepine skeleton and end up with useful drugs. All the things on the periphery here are useful, important drugs. And you can even try, this is a very unfortunate photograph of Corwin Hansch. Makes him look like he's wanted by the police in several states. He's a very distinguished chemist who has been working for years on quantitative structure-activity relations. In other words, harnessing the power of statistics to try and understand from a vast database what are the important features. And he tackled the so-called QSARs, quantitative structure activity relations of benzodiazepines. And after 22 pages of dense statistics, what comes out is this, which is almost exactly nothing, okay? In other words, even Corwin Hatch can't crack it. And this is a British expression, elephant gives birth to a mouse. So in order to make things worse, just when Corwin Hatch thought he was almost home, several new drugs have come out that act on the same binding site and have nothing to do with benzodiazepines in structure. And they're listed, the three from left here, and they're called the Z drugs. Zolpidem, Zalipron, Zopiclone, and that's Ambien, Sonata, and I forget the other one. You can see that the picture is getting bleaker and bleaker in terms of understanding how these things actually work. Now, a few years back, there was Villar, he's a quantum chemist, made an interesting observation, which was that there was a correlation between the activity of benzodiazepines and the energy of the lowest unoccupied molecular orbital. And this, I remember reading this and thinking, this is kind of weird and interesting. And then I forgot about it promptly for many years. Until, okay, let me explain why I thought that was interesting. The reason why the energy of the LUMO may be important is if you're interested in charge transfer, okay? If you have a charge transfer, these are two molecules that are used in chemistry as the exemplar of charge transfer. TTF is extremely electron rich, and TCNQ is extremely electron poor. Okay, so you mix them together, and they form a sandwich, in which the electron basically gets transferred from the HOMO of TTF to the LUMO of TCNQ, which happen to be at roughly the same energy, and this thing conducts. Okay, the electron. This is actually a room temperature metallic conductor. The solution has a metallic luster, as far as I know. So I thought this was, when I saw the Villar's idea about benzodiazepine, I thought this was interesting. But it was interesting for me in a slightly different context, which is to do with whether proteins are conductors or not. One of the fundamental tenets of biochemistry is that proteins are not electronic conductors, okay? They're insulators, okay? And you can make them conduct, but only under certain conditions. And these conditions were explored by a great man, Albert Szent-Györgyi. I assume some of you are familiar with him. He discovered vitamin C, got a Nobel Prize in '38, I think, for it, and then went on to do all manner of strange things. And he had his own lab at Woods Hole, and he found a millionaire that funded his research the rest of his life. Anybody here who wants to do that? My email was in the thing. He didn't believe in writing grants because he said, if I knew what I was going to do, I wouldn't be a scientist. Okay. So what he did was he studied the properties of protein chemically modified to make the electrons move. And how he did it was this. He took an ordinary protein, casein, albumin, doesn't matter, and you treat it with a compound called methyl glyoxal, which is also called pyruvic aldehyde for those who study biochemistry, and which is very closely related to the butter flavor in popcorn. It smells intensely buttery. And this thing is very reactive, and it binds to the terminal amino groups of one particular amino acid, lysine. And when it does that, it makes an imine. You will recall that the benzodiazepine ring has an imine in it. So we're homing in onto the real problem here. So when you do that, you find, this is what Szent-Györgyi discovered, you find the proteins go black. They treated with methyl glyoxal, albumin, which is initially white, goes a dark brown. And that, to a physicist, is immediately a sign of electron mobility. What's happening is the electrons are absorbing, the electron transitions are absorbing light in the visible range, so you've lowered the energy from the ultraviolet, which is where it normally lies, to the visible. And you can see these are spectra. Spectrum 3 is native albumin, 2 we'll forget about for the moment, and 1 is treated with methyl glyoxal. And this thing at the bottom is the wavelength. You see in the visible range it absorbs pretty nicely, which is why it's brown. Now, Szent-Györgyi, of course, had he in his day had DFT, software would have cracked everything and would have left nothing to us, but he didn't. So you can calculate things that he couldn't calculate. And one of the things you can calculate is the HOMO-LUMO gap, the excitation gap for untreated alpha helix with a lysine, and that's on the left, and that's 5 electron volts. That's way in the UV. And you put an imine on it, you treat it with methyl glyoxal, and you look at the energy, and it's 3 electron volts. So it's come down quite a bit. And if you look back at that graph, the DFT predicts pretty accurately the peak of the absorption at 3.61 EV, which is in the violet. Now, those observant among you will notice that excitation, that the absorption, sorry, of the protein here stretches all the way east of that, so that even though we correctly predict the 3.6, clearly there are things that are enabling the protein to absorb at much lower energies. And for a long time, I think that was completely unclear as to what could be going on. Why would it absorb all the way into 450, say? And St. George's point of view, which is quoted in this slide, was that electron mobility was a fundamental thing, okay? Now, no one these days would dream of using the word clumsy to describe macromolecules. That's, in biology, that's sacrilege, okay? And arguably, it is sacrilege, because macromolecules do extraordinary things. But he felt there was something missing. Now, the people's reaction to his work was, well, that's fine, but you've modified them with methyl glyoxal. That doesn't really happen, so who cares, okay? One word on that. Methyl glyoxal now is considered one of the primary agents of protein aging. So that when an animal ages, you get all manner of covalent reactions that happen between, let's say, sugars and other reactive species in the cell. And one of them is methyl glyoxal. Okay, so I just want to put this in the back of your head that actually St. George may have been looking at a physiological or pathological phenomenon rather than an artificial. But I think no one was interested because it seemed too artificial. Now, what happened recently, or when I say recently, I mean the last 20 years. When you get to my age, recently extends further back. Peter Kovacic, who's in his early 80s and still very active, took the whole idea of imines one step further by implicating iminiums. The imine group can be protonated and become an iminium. And Peter Kovacic has documented the effect of iminium-containing molecules in pharmacology and biochemistry all over the place. Now, why is this interesting? Well, because you can do this, and you can go from imine to iminium. You just protonate it out of charge. Now, what happens to the energies? We've seen that the unmodified thing, huge gap, no absorption in the visible. The modified imine, 3.6, let's say, but the iminium is 0.2 electron volts. Now, 0.2 electron volts, now we're talking. This is no longer the UV or the visible. This is in darkness, okay? So if you manage to put an iminium somewhere, you may have electronic transitions from, let's say, the valence band to the conduction band, so to speak, that happen in darkness. So what you've got yourself is a proper semiconductor. Okay. Now, Kovacic pointed out that this could be possible, but I don't think he's very fond of quantum chemistry software, so I had to do it myself. If you look at the electron affinity of benzodiazepines, you have the bare skeleton. The nitro group introduction gives it electron affinity because nitro is electron withdrawing. You add another fluorine, you get a bit more electron affinity. But if you put that extra one hydrogen atom on it, all of a sudden, the LUMO falls through the floor, and you end up with an electron affinity with respect to vacuum of 5.4 EVs. Now the 5 EVs is the magic number, because if, in order to take an electron from the vacuum into the molecule, you gain 5 EVs, that's basically valence band for solids. How do we know that? Because it takes 5 EVs to get electrons out of a solid. That's the photoelectric effect. So this is not just theory. There's plenty of evidence out there that benzodiazepines actually do this in real life. In fact, they're just about the only drug that can be measured in an electron capture detector directly without modification of the molecule. They have such a high electron affinity that you see them in electron capture detection. So why is this interesting from pharmacology standpoint? Well, because if you go back to the structure-activity relation, the one that's never mentioned is the fact that if you don't have the imine, the C double bond N on the left, if you saturate that, okay, the diazepines don't work. That is a fundamental SAR. So you have to have the imine and therefore possibly the iminium in order for it to work. There are two exceptions. I know these days that people will look things up and then email me, so let me forestall that. There are two exceptions. There are two perfectly good benzodiazepines. Don't start out with an imine, ketazolam and cloxazolam. They're prodrugs. They actually break down to the compound that has the imine before they start acting. Now, why is iminium really interesting? Well, it's interesting because all the drugs, all the Sonata, the Zolpidem, the Zaleplon, the Zopiclone, they all have an iminium with pKs in the right region. So this is beginning to look like it might actually do a bit more explaining. So can iminium cause charge transfer and therefore conduction in dark, in proteins? The St. Georgi charge transfer complex was covalent. The TTFTCNQ is non-covalent. So we're thinking of a non-covalent. We're thinking of basically the drug sticking to the receptor and causing electrons to jump from protein to the drug. You can actually calculate that. I mean, it's boring. You can watch these calculations roll for 12 hours. I do that all the time. I just sit in front of my computer and watch the iterations. But you can calculate an alpha helix. This structure on the right is an alpha helix with two phenylalanines, and you attach it to rohypnol molecule, and you calculate the HOMO-LUMO distance with and without the iminium. And without the iminium, just the imine, 2.8 electron volts. With the iminium, 0.19, more than a factor of 10. Okay? So, and this, what this tells you is that if benzodiazepine form iminiums, they're in a position to suck up electrons from proteins in such a way that the protein that gives the electron is probably going to conduct in the manner that St. Georgi discovered. Now, how do we know that this may be happening in real life? Well, you have to know where to look, is the answer. Because there's no direct measurements of conductivity of GABA receptors. Nobody's doing that, partly because nobody cares. But they've done it accidentally, and this is what I'm going to talk about. People discovered some years back that you could alter GABA receptors function by putting electrons, so to speak, in the outside medium, electron donors. They're called redox reagents. That if you change the redox state of the receptor, it would behave differently. I just want to say, by the way, that this discovery was made in Russia, in the then Soviet Union, in the late 70s by Pupi and Kazachenko. That their paper by Pupi and Kazachenko was utterly ignored for 20 years. That they got zero credit for discovering this very important and interesting part of pharmacology, and they're still getting very little credit. They're mentioned, they're sometimes cited and sometimes not. But this is a perfect illustration of how much good biophysics was going on in the Eastern Bloc that has just basically been forgotten. So what they do is they put drugs that push redox reagents act on a particular type of chemical bond in proteins, the disulfide group. And if you push, you can push them towards making disulfide bridges between two amino acids, or you can push them towards breaking the disulfide bridges. And what they find is totally remarkable. You're measuring the GABA receptor, and you put GABA, and it responds every time nicely. Then you put something that breaks disulfide bridges, and you get a bigger response. That's the DTT in the middle, the peak of the red bit. And when you remove the DTT, it goes back. And that's perfectly normal because disulfide bridges will spontaneously the two SHs will spontaneously reform to make a disulfide bridge. So you'd expect the DTT to be reversible. But then the fun happens. You put DTNB, which causes them to make a disulfide bridge, and you find that the effect of DTNB is reversible. Okay? Now, this is very strange. This is just not an isolated observation. The other group who did that and published it at the same time found exactly the same thing, almost the same trace. Okay? So why is this weird? Well, because the disulfide bridge they're working on is in the outside medium. It's actually known which one it is. There's only one, okay? And it's in a place called the cis loop of the subunits. So there are no electron donors in the outside medium. Every disulfide should be as a bridge, should not be as two SHs. So the fact that they should be fully oxidized, but when you put an external oxidant like DTMB, it acts transiently. The thing clearly re-breaks. Something is keeping those extracellular sulfides reduced. Okay? That something can only be electrons. There's nothing else that will work. And therefore, the electrons must be coming from inside the cell. Now, when I figured this out, I contacted Trevor Smart, who had actually done the experiments, and he said, you know, we always wondered why these things were switching back, but we couldn't figure it out. Biologists aren't used to thinking of electrons crossing membranes. So let's just take a step back here and look at how this circuit might work. You've got the benzodiazepine binding site is on the outside between two subunits. You have the cis loop, which is not far from the benzodiazepine binding site. In fact, it's extremely close. It's only a fragment. It's along one of the protein ribbons. And where are the electrons coming from? Okay, they're coming from inside. But you can't just give an electron to a protein out of the, in a random way. There has to be a binding site on the protein to accept electrons from an electron donor. So what I did was I went looking for for sockets, so to speak, in the primary sequences of GABA receptors. And this is a amino acid sequence. G is glycine, then anything, then either glycine or alanine, then anything, and then either glycine or alanine. So this GXG, XXG, or A is a diagnostic sequence for electron binding sites. Incidentally, there's nothing super duper mysterious about this. These are very small amino acids, the smallest side chains. So basically what you're doing is peeling off the insulator to expose the peptide backbone. Okay? The peptide backbone is probably what's conducting. So when you have glycines and alanines in this arrangement, the donor molecule can approach the backbone enough to give electrons to it. Okay? And you find this binding site in the alpha subunit, okay, of the type of subunits that are sensitive to benzodiazepines. So there's a very good indication that the socket is present in the right place. And the socket binds a soluble electron donor. So here's the electron circuit that I'm postulating exists in the GABA receptor. You have NADH giving electrons to one subunit. The electrons travel across to the benzodiazepine binding site, jump across, probably through the benzodiazepine, very likely, and reduce a cis loop, which then opens up and controls the function of the receptor. So what we're looking at here is actually not a lock and key. It's a field effect, a chemical field effect transistor. It's chemical because you need to protonate the benzodiazepine in order to make it work. Okay? And it's once it's turned on, it acts as a gate for electron flow between subunits. Now, how would you test this? Well, the thing about molecular biology is you can have fun. You can do all manner of stuff. You can mutate the NADH binding site, the electron socket. You put a big fat amino acid, it won't work anymore. Easy to do. I mean, on a scale of 1 to 10, 3, easy to do, okay? Not 10. You can measure electron flow through the protein during the experiment. If you have electrons flowing, hopping through, you should see a change in capacitance, basically. The protein becomes conducting. It's in an insulator. You should see that as a capacitance change. You could try ligands with a very, very high electron affinity. In fact, I'm thinking of some of the anesthetics have a very high electron affinity. It would be very interesting to see if you can trigger off, if that's the mechanism that they use to turn on the GABA receptor. And finally, you can mutate the electron path to the... So, in conclusion, next time you take, well, those of you who don't take benzodiazepines will be unaffected by this, but next time you take a breakfast of champions like this, when you're looking at these molecules in the form of pills, think of them as electronic components. You may be swallowing a bunch of transistors. This clearly, you would agree with me that this is 1.1, right? Triviality level. This is step one. I'm very concerned with doing things that are experimentally measurable because I'm not enough of a theoretician to have a good time with theory, okay? So this is step one, but consider the possibility that there are actually quantum effects within the molecule. People have demonstrated wonderfully interesting, I mean, since the beginning of molecular electronics, there's been an awful lot of talk about interference between different conducting paths of a molecule. I for one, would not be so surprised if somebody, based on this tomorrow, showed me that the reason some benzodiazepines work and some don't is because some have a constructive interference of electron flow through them and some have a destructive interference. This is very much step one. We don't know where this takes us. We don't know I don't know whether it connects in any way with what Stuart is saying. But what I'm saying is, here's a situation where we thought we had it pretty much locked down and no electrons, no quantum stuff was involved. And it turns out it may be that the level one quantum stuff is involved. Thank you very much. Yes, absolutely. It depends on pH in a completely normal way. The pKs of these things can be anywhere between, let's say, 2 and 8. Of course, pH, bulk pH is relatively meaningless. If your drug is bound next to an acid group, you're effectively dealing with a very high local concentration of hydrogen ions. So, yes, it is pH dependent, but whether it's pH dependent in situ is not known.
So the second question is, you gave this sequence of GX,
Yeah.
Is this kind of a universal statement? Or
Yes, let me just find the slide that that deals with that. No. Yeah, that's the one. Yes. The GXG-XXG is a universal, or not universal, but is a very common binding sequence. Stuart.
Yeah, thank you very much, Luca. Just to be clear, are you saying that the conduction is through nonpolar hydrophobic regions? Because I saw phenylalanine.
Okay.
Conjugated rings, so these are resonant ring structures that the electrons are going through?
Well, not really. If the conduction has to take place over the entire span of the protein, it won't find a, if you want, phenylalanine type network to do so. Very likely what's happening is conduction along the peptide backbone. And the reason for that is that the peptide bond itself is, of course, conjugated, but there are some interesting aspects to it, sort of a hyperconjugation, such that the it may be that there's a low conductivity all the way along the backbone. This is still pretty contentious, but as DFT techniques improve, excuse me, and as in particular, as the linear scaling DFT that enables you to calculate whole proteins comes on stream, we'll find out whether the electronic structure of proteins actually includes low-lying conduction band states directly.
How far apart would aromatic rings have to be to conduct organics?
I think Elizabeth can probably answer this one better than I can. I think the answer is it depends how fast. The tunneling phenomenon, of course, scales viciously with distance, okay? Exponential distance squared. So if you're only interested in 1 electron every 2 weeks, they can probably be quite far, okay? The thing about these receptors is that, as you saw, the time scale of reduction is tens of milliseconds, possibly. So, and 2 electrons every 100 milliseconds is not, is not a very stringent requirement for tunneling. That's a low tunneling current. So the answer is probably further than we think right now.
We were talking a little bit about this during the break, but how does the drug molecule find its way there? Does diffusion explain everything?
Yeah, I don't think this, I think diffusion explains, let me clarify this. Diffusion explains how it finds its way to the active site. Shape explains why it binds to the active site with high affinity, okay? But binding isn't everything. You can perfectly well have an antagonist. What we call antagonists in pharmacology are things that bind like mad and do nothing, okay? And in fact, flumazenil, which is the #1 antagonist for benzodiazepines, does not have an imine on it. It just has the right shape, okay? So the equation, the mental sort of association of affinity with efficiency is, of course, very easy to make. Pharmacologists don't make that mistake. They have another thing which is called efficacy. Okay? So something can bind like crazy and do precisely nothing because it has an efficacy of 0. The problem is once you've called it efficacy, you still haven't got a clue as to what is actually happening. Okay. Thank you.
Does effects occur in other types of drug interactions other than psychoactive drugs?
Other than psychoactive drugs?
Yeah.
Yes. If you read Kovacic's papers, he has found iminiums in all manner of strange places. I can't recall all the categories, but the answer is yes. Yes, absolutely. Thank you very much.